Okay, so in this video again, we will look at the thrombin, how thrombin is catalyzing the conversion of fibrinogen into fibrin. So fibrin is a long fibrillar protein, okay, sorry, fibrinogen, and this is how it will look like. And then thrombin, sorry, this is how it will look like. This is fibrinogen. It will have different chains. Then thrombin is a proteolytic enzyme. It will convert fibrinogen into fibrinopeptides. Okay, so it's got three chains. Don't worry too much about this because you're going to learn this next year as well. So it will remove small fragments from here and then form perfect chains here. And this is what is being converted into fibrin. Okay, it will look like this. So this is how thrombin is converting fibrinogen into fibrin. So again, there are so many different types of clotting factors, up to 13 factors. First one is fibrinogen, last one is stabilizing factor. Again, you can go through them later on, and they are different cofactors also associated, and they are also playing critical roles in clotting. So factor 8 is important for hemophilia. If it's affected, it's acting as a cofactor, it's not an enzyme. And factor 8, if it is deficient, then the patients will have hemophilia. That's a common disease in several individuals. Okay, how the negative feedback regulation is happening here. So we talked about for the platelets, how prostacyclin I2 and the nitric oxide was preventing unnecessary platelet activation. So similarly, the coagulation cascade should not go on in the body unnecessarily. So they had to be stopped through negative feedback regulation. So how the negative feedback for coagulation cascade is happening here. So we got again various molecules that can prevent the unnecessary activation of coagulation factors and the coagulation cascade. So the first important molecule is called antithrombin 3. This molecule, this is a protein which will attack the active form of thrombin because it's a multifunctional enzyme, and it will inactivate this thrombin. Therefore, the thrombin cannot convert fibrinogen into fibrin. Similarly, antithrombin will also attack and inactivate active form of factor 10 and active form of factor 9 and active factor form of 11. It will inactivate all these molecules. So this is an important negative regulator for blood coagulation. Similarly, there's a molecule, there is another protein called plasminogen. This protein will be converted into plasmin, and that's a blood clot dissolving enzyme. Plasmin is a blood clot dissolving enzyme. We're going to talk about that later on. So that's also important that we have in the body as well. Then protein C, this protein C will inactivate factor 5, active form of factor 5 and active form of factor 8. And then there's another protein called protein S that will accelerate the functions of protein C. So all these four molecules, these four proteins are really important negative regulators of coagulation cascade. Without these molecules, the body will clot unnecessarily all the time. Then we will have a lot of problems, and we don't want that to happen in the body. So this is what is happening. Okay, next stage. So the fourth stage of blood clotting. Now the blood clot is formed, platelet aggregate formed, then there's fibrin clot formed, fibrin clot stabilized. But the clot can't stay there forever. The wound has to heal and the body has to repair the damage, then move on. So we can't keep the clot for all the time. So this is when the clot retraction is happening. So retraction means it's basically retracting the clot or constricting the clot or squeezing out the clot. So it's shrinking the clot to enhance the wound healing. So when it's shrinking, it's bringing the wound edge together. Therefore, the wound edge will damage and the tissue repair will occur. So this is a post-clotting event. After the complete clot is formed, it's prevented, and after some time is gone, the clot retraction will kickstart. Okay, it's mainly induced by the activated platelets present in the blood clot. Okay, you need to remember that this is another major functions of platelets, activated platelets particularly. So activated platelets already aggregated and it's making the temporary clot. Activated platelets will provide a surface for blood coagulation. Similarly, activated platelets are important for clot retraction. Without platelets, the clot will not retract. So there is an important molecule called integrin present on the surface of platelets. Okay, and these molecules are driving the clot attraction through platelets within the blood clot. Okay, these are integrin molecules. Don't forget. Okay, so the clot will retract and it will squeeze out all the liquid, which is basically serum present within the clot. It would have entrapped. Now it will squeeze out. So this will help to bring the wound edges together and promote the wound healing. That's the whole purpose of clot retraction. Then the final stage is clot dissolution or fibrinolysis. The clot has to completely get dissolved and removed from the body. Okay, so this is what is called clot dissolution or fibrinolysis. So once the blood loss has been controlled and the vessel walls have been pulled together, blood flow must be re-established in order for permanent tissue repair to take place. And the process by which the clot dissolved is called fibrinolysis. So like coagulation, fibrinolysis is a series of steps controlled by activators and inhibitors. There is an important enzyme here called plasmin. That's the enzyme that's going to drive this process. So plasminogen is present in our plasma or in blood all the time circulating. This is an inactive form of plasmin. Okay. There is a factor called tissue plasminogen factor that's also present in the blood. Okay. After the clot is formed, this factor will convert plasminogen into active enzyme called plasmin. Now plasmin, it's an enzyme. And fibrinopeptides are the substrate for this enzyme. So it will bind to fibrinopeptides and break down all of them. It will just literally bind to the fibrinopeptide in the blood clot and chop them all into small fragments or small pieces. This is how the clot is getting completely dissolved. Okay, it's called clot dissolution or fibrinolysis. There is another protein called alpha-2 plasmin. It is an inhibitor for plasmin to control its activity locally. We don't want the plasmin to circulate all over the body because it will not induce the blood clot. It will prevent the blood clotting by dissolving the fibrinopeptide. So once its job is finished at the local affected site, the plasmin has to be completely removed. Okay, so this is what is happening in the last stage of blood clotting. That's called clot dissolution or fibrinolysis. Okay, what stops all the blood in the circulation from clotting? We talked about this before. There are two molecules controlling the platelet activation, four main molecules controlling the coagulation cascade through negative feedback regulation. So what are the other molecules, other factors controlling our blood from clotting within the blood vessels when we are healthy? When we have diseases, it's completely different thing. That's pathological conditions. But when we are healthy, what are the molecules controlling? So the first thing is healthy endothelium. We have healthy endothelium, which covers the collagen, which is present underneath the endothelium. Okay, and the endothelium is producing nitric oxide and PGA2, and it's also producing tissue plasminogen activator. We just talked about for fibrinolysis. So we know that nitric oxide and the PGA2 are inhibiting platelet function unnecessarily. Collagen is fully covered. Tissue plasminogen activator will be circulating in the blood all the time to monitor any plasmins. Okay, so next one is clotting factors circulate in inactive form. All the clotting factors circulating only in an active form. Only when there is an injury, these factors will be converted into active form, and that's when they are going to induce coagulation cascades at the affected site, not all over the body. And then blood flow. Blood is not stopping in one place. It's just flowing all the time. As long as the heart is beating, the blood is keep flowing in the body or within the blood vessels. So because it's keep flowing, it's carrying away all the factors without stopping in one place. Okay, so when the blood is hanging around in one place, they are likely to clot. So but the blood flow is just keeping them in motion all the time. It's just to keep carrying away. And then factors present in blood that inhibit clotting factors, particularly antithrombin 3, protein C, and protein S. Okay, so they are all present in there to prevent any unnecessary blood clotting. Then consumption of clotting factors at the site of damage. So wherever we have the damage, that's where the clotting factors will be consumed, because that's where it's produced in mass scale and it will be utilized at the defined site rather than going all over the body. And then finally, we do have a fibrinolysis using the enzyme called plasmin. That's going to digest any unnecessarily formed blood clots and then remove from the system. So these are the factors preventing any unnecessary blood clots within our physiological conditions when we are healthy. Okay, now we are going to talk about the diseases relating to blood clot. Okay, so what can happen to the blood? So if it is clotting unnecessarily in wrong place and wrong time, that's called thrombosis. That can affect the blood flow to main organs, for example, heart and brain, and ultimately it will lead to heart attack and stroke. So that should not happen. If we have diseases that's likely to cause thrombosis, then we need to take medications or proper treatment. Then the next thing what will happen is bleeding or hemorrhage. That's basically insufficient blood clotting or hemostasis. If the blood is not clotting normally, then we will bleed. The excessive bleeding is extremely dangerous and we will go to coma very quickly, because if you don't have a sufficient amount of blood, cells will not get a sufficient amount of oxygen and the nutrients, and then they will go to ischemia and that will cause coma. That's a pathological condition. So there are two pathological conditions associated with blood clotting. One is unnecessary blood clotting or excessive blood clotting. Second one is bleeding, which is reduced blood clotting. Okay, so thrombosis and increased blood clotting. So what's happening here? So again, as you know, there are two different types of blood vessels in our body. So one type is called arteries. The other type is called veins. Okay, so we do have a venous circulation and arterial circulation. Arteries will carry oxygenated blood from the heart to all the tissues. Okay, that's bright red and it's carrying oxygen and it's going to supply oxygen to all over the body to the tissues. Veins will collect the utilized blood or deoxygenated blood from the tissues and take it back to heart in order to send it to the lungs for gas exchange. Okay, so we got arteries and veins. The blood clot can be formed both in arteries and veins. If it is happening in the arteries, that's called arterial thrombosis. I'm talking about pathological conditions here. Okay, that's called arterial thrombosis. If it is happening in the veins, that's called venous thrombosis. Okay, you need to understand the difference here. Both are pathological conditions. In normal conditions, the blood clot has to happen only at the damaged site. But in arterial thrombosis, blood clot can happen within the arteries. In venous thrombosis, blood clot can happen within the veins. Okay, so we talked about these stages before. So basically, if there is an injury, collagen will get exposed, platelets will get activated, clot is formed, then the coagulation cascade will make fibrin, and that will trap everything, make the clot. So here there are two important things. So one important aspect is platelets, the platelet plaque formation or platelet activation and aggregation. The other one is called coagulation. Okay, so this is really important here. Carefully understand this. So platelets will play major roles in arterial thrombosis, because in arterial circulation we will have a high blood flow. In venous circulation we will have a slow blood flow. So the flow rate will be very high in arteries and it will be slow in veins. Okay, so platelets are playing major role in arterial thrombosis and the coagulation will play minimal role here. Okay, if you look at venous thrombosis, coagulation will play a major role and the platelets will play minimal role. Yeah, you need to understand the difference. In arteries, it's the thrombosis is mainly driven by platelets. In veins, thrombosis is mainly driven by coagulation cascades. In arterial thrombosis, examples for arterial thrombosis are myocardial infarction or heart attack, and ischemic stroke, peripheral arterial diseases. For venous thrombosis, best example is deep vein thrombosis, which we will talk about again next year in too much detail. So these are the main reasons why we are taking different anticoagulant drugs. Okay, so if somebody is suffering from arterial thrombosis, classic examples, heart attack and stroke, they will likely to take antiplatelet drugs. These drugs will prevent the unnecessary activation of platelets. If someone is suffering with a deep vein thrombosis, that person will get anticoagulant drugs, for example, warfarin, that will prevent the coagulation cascade. Okay, guys, please carefully understand here. Okay, arterial thrombosis mainly driven by platelets, and therefore they will take antiplatelet drugs. The coagulation cascade will play minimal roles here. Venous thrombosis is mainly driven by coagulation cascade, and therefore they will take anticoagulant drugs, and the platelets will play minimal role here. So this is the main difference. If you understand this, next year thrombotic diseases will be completely easy for you. So here you can see thrombosis or increased clotting. So for example, in arterial thrombosis, that's a heart attack and stroke, and this is how it will look like. So here you can see it's a massive atherosclerotic plaque. It's basically liquid accumulation within the blood vessel. At one stage, it will get ruptured. When it's rupturing, it will expose the collagen and all the other factors, and therefore blood clot is completely formed, and that completely sealed the area. It already had a narrow lumen because of the lipid deposition, and now blood clot is formed, and that will completely seal the blood vessel. Therefore, there won't be any blood flow in the downstream. If this is happening in the coronary artery, that will lead to heart attack. But if this is happening in carotid arteries, that will lead to stroke. So in our lab, we use different animal models. For example, laser induced thrombosis. So within the animal, you can open up any blood capillaries and fire with the laser, and you can continuously monitor the thrombus formation. This is how the thrombi will be formed. Okay. So how the venous thrombosis is formed. So this is mainly formed in the deep veins. So you can visualize this using X-ray. Okay, if you use X-ray, you should be able to visualize thrombosis like this, like a dark color, stained molecules. Stained blood clots. So it's really important to look at this. Okay, so why this is happening? This is mainly happening due to inappropriate activation of coagulation within the veins. Again, platelets will play a role here, but very, very minimal roles. It's mainly driven through blood coagulation cascades. So there are three important factors here. So it's normally called as virtuous triad. So the first factor is blood stasis, or hemodynamics is affected here. So imagine you are taking a long haul flight. You are traveling to Australia from London, okay? Non-stop flight, 18 hours you are traveling. So, and if you are not walking around the cabin, just sitting in your seat, then it will cause blood stasis. You need to remember the vein blood flow is mainly driven through muscle action. So the muscle has to do some action in order to keep the blood flow to move as much as possible. Okay, otherwise, if there is no muscle action, if there's no movement, the blood will start to get stasis or hang around behind the venous valves. So when this is hanging around for a long time, slowly the coagulation cascade will get activated because the blood flow is keep moving the coagulation factors. But if the coagulation factors are not moving around continuously, then they are hanging around here for a long time. If they are hanging around for a long time, coagulation cascade will get slowly activated. It's not an instant process. It will take several hours, and sometimes it can take even days to make this. Okay, so this is happening, and then slowly it will make a small clot. Okay, and this clot, because it's not really stable, it can get pinched off. When it gets pinched off or broken down, that's what is called embolus. That whole process is called embolism. Now this clot can straight away go to heart. From heart, it will be pumped into the lungs, and it will cause a serious problem. That's why deep vein thrombosis followed by embolism is a really serious problem. So we need to take proper treatment quickly. So during a long haul flight or a hospital stay, if somebody is admitted in the hospital for several days, the blood stasis will occur. That's why they need to be given anticoagulant drugs in order to prevent the blood stasis. Even if the stasis is happening, prevent the blood coagulation or blood clot formation. The next one is endothelial damage. If the endothelium getting damaged within the veins, okay, unnecessarily, then that will slowly activate the blood clot, and that will also cause this problem. Okay, the next one is a hypercoagulability of the blood. Some people, again, due to increased level of platelets or increased level of specific coagulation factors, they are prone to clot very quickly. So this is what is called the hypercoagulability nature of the blood. If they are coagulating very quickly, then they are prone to make the clot if they are not active even for a short while. So these are the important factors controlling or inducing the coagulation within the venous circulation. There's a minimal role for platelet, as I said before, and the embolism can happen that can pinch off the blood, that can pinch off the blood clot, and it will enter into the heart, and then it will pump back into the lungs, and it's a serious problem. Okay, so we can talk about the remaining things of this lecture in the next video. Thank you.